Today I'd like to follow up on our previous discussion of the major macromolecules, biomolecules in nature, and look at how those molecules then form cell structures and cells themselves, which are the basic unit of life. And so before I actually get to the cell itself, I want to discuss a few topics that are related. We're going to be discussing plasma membranes, and plasma membranes make up the outer boundary of the cells and a lot of internal structures in cells. And actually, before I get to plasma membranes, there are a couple of other terms that I need to quickly discuss. And so one of those is, or would be a group of terms, which are synonymous for the word polar. Now, we learned about polar covalent molecules before, large molecules that have regions of positive and negative charge. Even small molecules, they can have regions of positive and negative charge. But if something is polar, that means that it's water soluble. It readily dissolves in water. Another term, or other terms that we use for that, would be that it is fat insoluble, so it will not dissolve in fats or lipids. That in turn makes the molecule hydrophilic, meaning that it loves water, and also lipophobic, meaning that it fears or doesn't like fats, will not dissolve in fats. So these are all interchangeable terms. I'm probably going to use them all. I'm not trying to be deceptive or anything when I use them, but depending what author you look at, any of these terms could be used for polar or water soluble. And then conversely, nonpolar molecules are water insoluble, do not readily dissolve in water, because these would be primarily fatty substance, and so they are fat soluble. And they are hydrophobic, again meaning that they fear water, don't dissolve in water, or are lipophilic, meaning they love fats or will dissolve in fats. So using those terms in, or looking at the actual structure of a plasma membrane then, the basic structure of a plasma membrane is that it is composed of a double phospholipid layer. So we'll be looking at two layers of phospholipids. And you may recall that phospholipids have areas that are water soluble. So they have so-called water soluble heads, and then water insoluble fatty acid tails. And so again, you may recall in our discussion of phospholipids that the tails form inwards, and the water-soluble heads, hydrophilic heads, will be found on the outside. So a typical plasma membrane then is composed of a biphospholipid layer, two layers of phospholipids with proteins embedded in that layer. And those proteins can have a variety of functions that we won't really get to now. But again, this is very typical. It doesn't matter if we're talking about a human being, a moose, an elk, or a bacterium. This is the basic structure of any plasma membrane. Now, plasma membranes allow some things to pass through, but not everything to pass through. So when we talk about plasma membranes, we describe them as being semi-permeable barriers or semi-permeable membranes. And that's usually just abbreviated in SPM. And so looking at examples of particles that can diffuse through these, again, they have water insoluble interiors, water soluble heads. And so this means that lipid soluble products can pretty easily move through the membrane. If they can get past the heads. Small molecules like oxygen, if they do not have a charge, can also pass through the membrane. However, polar molecules and also charged particles have problems getting past the water-soluble heads, and so tend to be unable to pass through the membrane. So again, some things can pass through, some things cannot. That's what defines it as a semi-permeable membrane. Now, larger particles, if they can't pass through that biphospholipid layer, it is possible that they may be able to pass through pores that we find in the membrane, or there may be so-called transport molecules that can move those particles across the membrane. So again, not everything is able to get past across the membrane, so it is semi-permeable. All right, so let's go ahead then, and also I want to briefly discuss solutions. So when we look at cells, and the membranes within cells, they're basically separating the cell into different kinds of solutions. And so a solution is a homogeneous mixture of ions or molecules of two or more substances. And when we look at a solution, the solute will be the relatively less abundant substance, and the solvent will be the relatively more abundant substance in a solution. Now, in life, and the term solution can apply to liquids or gases. And in living solutions, we're normally talking about aqueous solutions. And so in aqueous solutions, we're going to be looking at two components here. The solute will be dissolved solids or gases, and the solvent is going to be a liquid component. It's going to be water. So in aqueous solutions, solutes will be dissolved in water. And basically in cells, then we're looking at complex solutions of various types. And I'll be coming back to solutions at another time, but next then I want to actually get into the cells and the cell structures themselves. So there are two major types of cells, prokaryotic cells and eukaryotic cells. And prokaryotic cells get their name from the fact that they lack a true nucleus. So pro means before, karyo is a reference to nucleus, so they lack a true nucleus. And what that means is that the nuclear material or the genetic material is not encapsulated within plasma membranes. It's basically free within the cell. And they also tend to lack membrane-bound organelles, complex organelles. They have small ribosomes, which we'll talk about later, and very characteristic flagellae that are unique to prokaryotic cells. And then also prokaryotes tend to be very small, typically less than 10 micrometers. A micrometer is a millionth of a meter. And so a very typical size for a prokaryote is 2 or 3 micrometers. So looking at prokaryotic cells, working from the outside in, the most outermost structure that you would run into on a prokaryotic cell is what's known as a capsule. Not all prokaryotes have a capsule, but when a capsule is present, it's primarily composed of lipids, sometimes what we call glycoproteins, sugar and proteins combined. And the capsule in some prokaryotic organisms, such as bacteria, and when we talk prokaryotes, we're talking about bacteria and organisms called archaeans. But the capsule a lot of times is associated with disease-causing bacteria. So the capsule itself may contain toxins that are potentially cause disease, or it may help the organism avoid detection by the immune system. Internal to the capsule or associated with the capsule and the cell wall are little fibers that are known as pili. And some of these pili, also called fimbriae, the function of the pili is that they can help connect prokaryotic cells together or attach prokaryotic cells to surfaces. Some pili also have a function in transferring genetic material from one prokaryotic cell to another. So that's another possible function. Anyway, these pili are important because it helps the bacteria attach to surfaces, and we see these so-called bacterial mats in the hot springs throughout Yellowstone. So you can see that there can be a variety of different types of prokaryotic organisms forming these prokaryotic mats, and this is something we'll come back to later in the course, because depending on the kind of spring you're looking at, the color of the mat that you're seeing can tell you what kind of organism it is, what type of prokaryotic organism it is. But you can see also that some of these bacterial mats are just absolutely beautiful in terms of the coloration that you see and the patterns that you see. And again, this coloration can be very dramatic. Also, when you combine that with minerals that are dissolved in the water, the hot springs in Yellowstone can just be spectacularly beautiful. But again, a lot of that coloration is due not just to minerals in the water, but to the living organisms that are there. Okay, now moving inward then, interior to the capsule is a cell wall. And the cell wall is composed largely of what are known as glycoproteins, a combination of sugar and protein. And the cell wall, as well as the capsule, I should mention, are highly permeable. All kinds of molecules can move freely through the capsule and the cell wall. And we really don't consider those part of the living cell. They're kind of external to the cell, if you will. And in some of the prokaryotes, you'll have a specific material called peptidoglycans that may be present, and it can be a thick layer or a thin layer of peptidoglycans. But again, we consider this as being a structure that's external to the living cell itself. And again, it's protective. It also prevents the cell from swelling up and rupturing under certain conditions. Interior to the cell wall, we have the cell membrane. And so the cell membrane is the outermost boundary of the living cell. Again, it's a classic plasma membrane composed of a double layer of phospholipids with different types of proteins embedded in that. And those proteins, again, can be enzymes. They can be pore proteins that allow materials to move back and forth through the cell, and other types of materials as well. But that is the semi-permeable barrier. So that's really where decisions are made about what's going to enter the cell, what cannot enter the cell. Now, associated with the cell membrane and cell wall are flagellae, and the bacterial flagellae are unique to bacteria and other prokaryotes. I guess I should really call them prokaryotic flagellae. They're going to be different than what we see in our own cells. And so they have kind of a unique structure at the base. They have a rotor type structure, and so these bacterial flagellae will spin around, kind of rotate around, and that is a different kind of mechanism than what we see in higher cells, the eukaryote cells. Moving into the cell then, we find the genetic material. Prokaryotes tend to have a single large circular chromosome, and then that's compacted into a mass known as the nucleoid. So again, this is very different than eukaryotic cells we'll talk about later, in that there is no protective membrane around the nucleoid or the chromosome in this case. Also, the chromosomes tend to be what we call naked. They're not protected by proteins in any way. And we'll again talk about genetic material a little later in the course. Now, in addition to the chromosome, the single large circular chromosome, which is jammed in there also can be smaller loops of DNA, and we call these plasmids. And so plasmids are materials that can actually be moved between and transferred from one bacterium to another, or sometimes exchanged between bacteria. And this is how things like bacterial resistance, antibiotic resistance, is transferred from one cell to another. Those will be part of a small loop of DNA called plasmids, and then one bacterium can transfer those to another by means of the sex pili. Other structures that we find in the cytoplasm of the prokaryotic cell would be ribosomes. Ribosomes are structures that make proteins. The ribosomes themselves are composed of a material called RNA, as well as proteins as well. So anyway, ribosomes make proteins. And again, with prokaryotes, there's a specific type of ribosome that's unique to prokaryotes. And then also within the cytoplasm of prokaryotic cells, there can be additional membranes, just loose membranes within the cell. And some of these membranes are related to photosynthetic processes, and we'll learn more about photosynthesis later in the course. And then others are what we refer to as respiratory membranes, in which the process of cellular respiration is carried out with these membranes. But we'll learn more about those processes, photosynthesis and respiration, a little bit later in the course. Now, that brings us into eukaryotes, eukaryotic cells. Eukaryotic cells are much more complex. Eukaryotic cells, what the word is trying to tell us about them, the prefix EU means true, karyo means nucleus. So these are cells that have a true nucleus. And what defines a true nucleus is that it has a nuclear envelope surrounding it. The nuclear envelope is composed of two plasma membranes. So each plasma membrane is composed of a bilipid layer with proteins embedded, and the nuclear envelope is actually two plasma membranes. So also, eukaryotes, instead of having a circular chromosome, have a number of linear chromosomes. So they're not circular, they're just basically linear in structure. And also they're composed of a combination of DNA and protein. They possess membrane-bound organelles, and we'll talk more about that in a minute, basically meaning they have small structures within that are composed of membranes. And they have more complex ribosomes, larger ribosomes, so-called eukaryotic ribosomes, as well as eukaryotic flagellae and cilia. And also eukaryotic cells tend to be much larger than prokaryotic cells, greater than 10 micrometers. I would say 100 micrometers is kind of a typical size. Okay, so looking at first an animal cell, again kind of working from the outside in, you can see initially here again there's a lot more structure here, a lot more complexity, but again we would begin with the cell membrane or plasma membrane. The cell membrane is a plasma membrane. The cell membrane is the outermost boundary of the cell, and again semi-permeable barrier. And you'll notice that animal cells lack a cell wall. So here we don't have any external structures like a capsule or a cell wall. The outer boundary of the cell is the cell membrane, also referred to as a plasma membrane. And again, it's a classic plasma membrane, bilipid layer with proteins embedded. And there may be additional proteins associated with the plasma membrane in eukaryotes. And these are what we call structural proteins that help give some rigidity and structure to the cell. So we refer to these as the cytoskeleton. So the cytoskeleton will be just interior to the plasma membrane and give a little bit of rigidity and structure to the plasma membrane. Also, the cytoskeleton can act as a kind of like a tracking system. So particles can be attached to parts of the cytoskeleton and then be transferred from one part of the cell to the other. Now, the major structure within the cell, of course, is the nucleus. And the nucleus is composed of a nuclear envelope, and within that you'll find chromatin, and chromatin is a combination of DNA and protein. The genetic component of a chromosome is the DNA. And then also you'll notice that in the interior there is a dark structure here, and that is a concentration of another type of genetic material known as RNA. And so the nucleolus is this dark structure, and the nucleolus is composed of concentrations of RNA. And we'll learn more about the function of RNA later in the course. Now, in looking at the nuclear envelope then, again, the nuclear envelope is composed of two plasma membranes. There's an inner membrane and an outer membrane. And this outer membrane, as you can see, actually folds out to produce a channel system that runs through the cytoplasm. And we refer to this channel system that's an extension of the outer membrane of the nuclear envelope as the endoplasmic reticulum. Some endoplasmic reticulum has ribosomes embedded in it. Ribosomes are structures that make proteins, just as they do in prokaryotes. But when we see ribosomes embedded in the endoplasmic reticulum, we refer to that as the rough ER, or rough endoplasmic reticulum. And so the rough endoplasmic reticulum is a place where proteins are made, and again, it's a channel system through the cell so that products can move from one area of the cell to another. Again, also we have the chromosomes there composed of DNA. DNA wraps around clusters of proteins called nucleosomes, and then the nucleosomes are compacted, and this again also forms the chromosome. So we describe chromosomes as being composed of chromatin, but I want to emphasize that the genetic component of chromosome is DNA. And just again showing you the appearance of chromosomes again. Okay, again, continuing on with our idea of the endoplasmic reticulum then. So the endoplasmic reticulum is an extension of the outer membrane of the nuclear envelope. And if there are ribosomes associated with it, it's rough endoplasmic reticulum. And then there are also extensions of the rough endoplasmic reticulum that do not have ribosomes associated with it. And we refer to that as smooth endoplasmic reticulum. And again, this is another view of that, looking at the extension of the nuclear envelope, forming rough ER and smooth ER. So ribosomes, again, make proteins, can be associated with endoplasmic reticulum. And ribosomes have two parts to them, a large subunit, a small subunit. These are larger, more complex than what we see in prokaryotic cells. So they're unique ribosomes for both prokaryotes and eukaryotes. Next, Golgi apparatus. The Golgi apparatus, sometimes called the Golgi bodies, generally associate with endoplasmic reticulum. And so the relationship here is that there are products that we'll find within the endoplasmic reticulum. And so tiny vesicles, tiny spheres of membrane will pinch off. And they will carry the products from the rough ER, and then they'll fuse with the Golgi apparatus. And so then these products then will be modified within the Golgi apparatus. And eventually, vesicles pinch off of the Golgi apparatus and either move to the plasma membrane, cell membrane, and spew the contents outside the cell, or in some cases, Golgi will actually be producing certain types of organelles that we'll talk about in a few minutes. So again, the Golgi apparatus then is basically a processing area formed from stacks of membranes. And again, products will move from the rough endoplasmic reticulum through vesicles, fuse with the Golgi, be processed, modified in the Golgi apparatus, and then either packaged in vesicles for export outside the cell, or these vesicles will actually form organelles within the cell. Now, one of these organelles that are formed by Golgi apparatus are lysosomes. And so lysosomes are tiny vesicles that contain digestive enzymes. And so lysosomes then can actually destroy organelles within the cell. If you have, say, a mitochondrion that isn't functioning well anymore, lysosomes will fuse with those, digest them, and destroy them. Also, when cells die, the lysosomes will rupture, and this will initiate digestion and decomposition of the organism. And again, this is kind of showing us how a lysosome can associate with vacuoles or other structures and then digest whatever is within that particular vacuole. So the term vesicle and a vacuole I'll come back to in a minute, but a vesicle is just a small sphere of plasma membrane, basically a container. You could think of it like Tupperware for the cell. And then also a vacuole would just be a larger vesicle, just a large Tupperware for the cell. But basically the cellular Tupperware is composed of plasma membranes. So if you want to move something around in the cell, you package it in a plasma membrane and then move it to another part of the cell. Another organelle that is formed by the Golgi apparatus, so some of those vesicles that don't move to the cell membrane but stay within the cell or what we call peroxisomes. They're part of a larger class that are called microsomes. So microsomes would just be small vesicles that contain specific types of enzymes. Peroxisomes specifically contain something called catalase, which helps digest hydrogen peroxide, which is potentially deadly to a cell. And hydrogen peroxide builds up in metabolic processes. Mitochondria are organelles that produce a form of energy that's useful to the cell. So they're referred to as the powerhouse of the cell. They produce a molecule called ATP. And ATP is required for energetic activities in the cell. So when our enzymes need energy, they need that energy in the form of ATP, and mitochondria produce ATP. And mitochondria are interesting in that they have two plasma membranes, an outer membrane and an inner membrane. The inner membrane is highly folded. This helps increase surface area, and so it's in these inner membranes that a lot of the reactions that produce this ATP are carried out. And also mitochondria are thought to have evolved from prokaryotic organisms. They have plasma membranes that are a little more prokaryotic in structure. They also contain their own ribosomes, which are prokaryotic ribosomes. They have their own complement of DNA, and they can divide independently of the cell. So mitochondria, again, used to be prokaryotes living within eukaryotic cells and have evolved to the point where we are codependent on one another, and so are now just an organelle within that eukaryotic cell. And then the centrosome is a structure which aids in cell division in animal cells, so it helps the cell divide properly. Plant cells do not have centrosomes, but animal cells have them and require them for cell division. And when you look at the centrosome, it's formed from 9 doublets of tiny tubules called microtubules, and we find these centrioles at right angles to one another. So the centrioles are 9 triplets of microtubules at right angles to one another. And these help make more microtubules. We don't know exactly how they do what they do, but again, they are required for cellular division. And then as I mentioned before, vacuoles are just large membrane-bound structures. They're basically for storage, typically, within a cell. And then vesicle would just be a small vacuole. And there are some vacuoles that have specialized functions. In unicellular organisms, there are things called contractile vacuoles. And what contractile vacuoles do is that in a cell like this, they may be gaining water and swelling up with water. And so this vacuole will swell with water. Then it contracts and squirts the water out. So it prevents the cell from bursting. So contractile vacuoles help cells from accumulating too much water and eventually bursting. The cilium or a flagellum is associated with some cells. It helps with movement of the cell itself or movement of particles and fluids over the cell. And so we see flagellae associated with spermatozoa, for example. Also, the cells that line a respiratory tract will have cilia associated with these. And basically, cilia are short, flagellae are long, but structurally they're the same thing. And when you look at the cross-section of a cilium or flagellum, what you see are 9 doublets of microtubules with a single doublet centrally located. And then also at the base of any cilium or flagellum, there is what's called a basal body. And a basal body has exactly the same structure as a centriole. And centrioles are part of centrosomes. And so what happens in the eukaryotic cilium and flagellum is that these microtubules will slide past one another. So basically they elongate and come back. And the basal body somehow controls the beating of the cilia and flagella. And again, we don't know exactly how they do that, but they are found at the base of cilia and flagella and required for the functioning of cilia or flagella. So again, instead of a spinning rotor at the base, this is eukaryotic flagellae and cilia work by sliding of filaments one over the other. Now, looking at a plant cell and contrasting that with an animal cell, we'll see a lot of the same organelles, and I won't review all of those, but what is different about a plant cell is that plant cells have a cell wall. So external to the cell membrane or plasma membrane, there is a cell wall. In plants, it's composed primarily of cellulose. Sometimes it's reinforced with other molecules like lignin and so on. And in addition to that, they have other organelles, which are chloroplasts. And chloroplasts in some ways resemble mitochondria in that they are composed of two plasma membranes, outer membrane and an inner membrane again. And the inner membrane is highly folded into complex structures. We'll learn more about this later, called thylakoids. And again, what this does is increase surface area so that when light strikes these thylakoid membranes, that's where photosynthesis is initiated. So the role of a chloroplast then is for photosynthesis. Now, not all of these plastids then are involved in photosynthesis. So so kind of a generic term for a chloroplast is a plastid. So some of the plastids do contain chlorophyll and are involved in photosynthesis. That would be the chloroplast. But if you're a cell that's making up part of a flower, for example, you might be storing pigments that account for the color of the petal. And then also if you're in a root, what will happen is that starch will be stored in that plastid, and so it's called a leucoplast. The prefix leuco means white. So in other words, if it can't be used for photosynthesis, it'll be used for something else. And then also most plant cells have a large central vacuole called a tonoplast, and that helps maintain internal pressure within the cell, helps support the plant. And you've probably seen plants that are dehydrated, they'll wilt and tip over, and when they're well hydrated, they'll stand up erect, and that has to do with how much water is contained within the tonoplast. And then also plant cells have connections between the cells. These are known as plasmodesmata. And so that actually the cytoplasm of one plant cell is continuous with the cytoplasm of another plant cell because of these plasmodesmata. So summarizing then, plant cells then lack a cell wall, they lack plastids, they lack plasmodesmata, although they actually have their own version of that. And they also possess centrosomes and centrioles, and of course those are required for division in animal cells. Plant cells, in contrast, possess a cell wall, whereas animal cells do not, possess plastids, chloroplasts, chromoplasts, or leucoplasts, possess plasmodesmata so that there's communication from one cell to another, and lack centrosomes and centrioles. So again, as we look at the Yellowstone ecosystem then, we have a lot of unicellular organisms, some of which are going to be prokaryotic and some of which are eukaryotic. And then we have a number of multicellular organisms, plants, animals, and so on, which are composed of tissues formed by eukaryotic cells. And that's something we'll be exploring in future modules. See you next time.